Today we are going to learn a little bit more about life stage PBPK modeling. So first, I would like to say that at Cytovation we develop several tools over the past few years on forward dosimetry, reverse dosimetry, IVIVE. In 2019, we did a work for the EPA, for a regulatory submission for the EPA, and we created a specific application for this project that is based on PLETHEM. So here you can see how the application is looking like. So you can download the chemical of interest. So here it's for pyrethroids, and I will explain the case study right after. But you can select basically the pyrethroid of interest. You can run some generic simulations, also some multiple exposure scenario that are predefined in the application. And you can run some simulation to look at concentration in different tissues, especially in the target tissue, which is brain for pyrethroids. This application also has the capability to do Monte Carlo simulations and also reverse dosimetry. If you're interested in this application, you have a link and you can ask me. I'm Marjory Moreau, the Associate Director of the Computational Division at Cytovation. So just send me an email and I can send you the link. So there's potentially altered sensitivity in early ages. So we know that due to rapid changing, there are differences in sensitivity of children and adults to the effects of chemicals. But we know that using animal data is also problematic because maturation dynamics are species dependent, and also exposure levels between animal studies and human environmental exposure are different. So what's the role of PBPK modeling in early age risk assessment? So you know with my previous training that the primary role of PBPK modeling in risk assessment is to define the relationship between an external dose and an internal dose. So PBPK models provide a tool to predict the internal exposure in the target tissue in early ages in comparison to adults. So life stage modeling can be supported by what I call IVIVS, which is in vitro to in vivo scaling to address early life sensitivity. So there's two key determinants in early age pharmacokinetic. First, it's the dynamics of growth and maturation physiology. We all know that at different ages, our body weight is going to change, our cardiac output, the volume of our tissues or organs, and also the blood flow. But there's also changes in biochemical parameters across ages. So it's really important. And what is important to understand too is that there are limitations due to challenges in obtaining pediatric pharmacokinetic parameters. So that's why most of the time we are going to use alternatives like in vitro experiment. So here you have an example of the development of a life stage PBPK model. This life stage PBPK model allows us to have age-specific internal dose in plasma or target tissues. But to populate this life stage PBPK model, we need different types of data. Like I said previously, we need some age-dependent physiological changes. We also need age-dependent biochemical changes, and we know we need an age-appropriate exposure. And these physiological parameters, they have been defined pretty well in the literature. But for the age-dependent biochemical changes, it all depends on the chemical of interest, and most of the time it requires in vitro data-based parameterization. A read-across can be employed to develop the generic PBPK model for chemicals. We all know that for chemicals, most of the time we don't have human pharmacokinetic data. So as a proof of concept, the first step is to develop an IVIV-based PBPK model in rats or mouse, but in a species that is not human. Extensive in vitro metabolism parameters and in vivo kinetic data can be developed for different chemicals and used in the rat model to recapitulate the in vivo pharmacokinetic results. The IVIV approach used successfully in rat modeling is then similarly applied to develop the human life stage PBPK model that can be used to predict target tissue concentration in adults and children. So the generic model usually incorporates, like I said, age-dependent physiology, as well as maturation profiles of enzyme like carboxylesterase, for example, or cytochrome P450 in plasma and liver. So here you can see what I was saying previously. The physiology changes during growth can be the body weight, for example. So you can see here the evolution of the body weight across ages. But the tissue blood flows can also vary during our life stage. So, for example, the liver blood flow is higher around 20 years old, but then is going down. Same thing for the brain. The brain blood flow is going down as we are aging. Here it's an example of age-dependent changes in metabolism. So maturation profile of enzyme can vary, like I said in the previous slide, and you have a perfect example here with the maturation of some cytochrome or carboxylesterase across ages. So it's really important to take this into consideration when we do a life stage PBPK model. So let's look at the case study, which, like I said, was a submission that we did to the EPA in 2019. So pyrethroids, they are neurotoxicants with the primary mode of action through interaction with neuronal voltage sensitive sodium channels. We know that parent compound is the toxic entity, and the metabolism of pyrethroids is mainly by CYP and esterase, and that's how pyrethroids are eliminated, metabolized and eliminated from the body. So the initiation of the project was concerns for potentially higher sensitivity to pyrethroid exposure in juvenile rats that came from observed increase in high-dose lethality in neonatal rats. These observations in the rat could be the results of either or both age-related pharmacodynamic and pharmacokinetic differences. So the hypothesis was that juvenile rats have limited metabolic capacity that is largely responsible for the elevated internal exposure and increased neurotoxic effects of deltametrine following the high pyrethroid doses in rodent toxicity studies. But this still led to concern over potential differences in sensitivity between children and adults to pyrethroid neurotoxicity in humans. Here, it's an example of species and age differences in pyrethroid metabolism. So on this figure, you can see the x-axis is on the top, the rat hepatic clearance, and on the bottom line, it's the human hepatic clearance. And the first bar is for juvenile or 1 year old in human, and the second set is for adults. And in light blue, we have cytochrome, and in kind of purple, we have the carboxylesterase. So we know that hepatic cytochrome P450 and carboxylesterase are important metabolizing enzyme for detoxification and elimination of pyrethroids in both rats and humans. But developmental changes in the expression of this enzyme between rats and humans, metabolic clearance of pyrethroids appear to increase slowly but steadily after birth in rats, whereas xenobiotic metabolic competency develops more rapidly after birth in humans. So it's a huge difference. Significant species differences also exist in the relative importance of some CYP and carboxylesterase enzyme in pyrethroid metabolism. So here, for example, like I said, the top figures are in rats and the bottoms are in human. Again, x-axis represents juveniles and adults, or in human 1 year and 25 years old. And the y-axis is the hepatic intrinsic clearance in the liver. So here we can see the differences between the enzyme metabolizing pyrethroids. So CYP in blue again and carboxylesterase in purple. So we can see that in rats, top lines, cytochrome are the major metabolizing enzymes for deltametrine and CPM, cispermetrine, with always a higher intrinsic clearance in adult rats. For TPM, so transpermetrine, carboxylesterase are more involved in the pyrethroid metabolism. Now, when we look at humans, the bottom lines, we can see that carboxylesterase are the major enzymes metabolizing pyrethroids, and the difference between children and adults is less pronounced compared to rats, even if intrinsic clearance is still higher in adults. So when we look at the rat study showing that juvenile rats are more sensitive, it's important to characterize the basis for the greater sensitivity in younger rats and assess its relevance to humans. Such observations could be the results of age-related pharmacokinetic differences, as it is known that species differences exist in pyrethroids metabolism and its ontogeny. So now, what are the risk assessment applications of the PBPK model? So the Food Quality Protection Act assumes a special susceptibility of children to pesticides by using an additional tenfold safety factors unless there's adequate data available to support a different factor. So a few years ago, the EPA reduced the tenfold to a threefold that account for PK differences between ages. But is this threefold adequate, inadequate, or conservative? So to answer this question, a generic life stage physiologically based pharmacokinetic model based on modern parameterization approach of in vitro to in vivo extrapolation with the focus on supporting risk assessment for potentially sensitive ages was developed. So let's take a look at the method that was used. So here we used a read-across strategy, like I've explained in one of the previous slides. So as a proof of concept, the first step was to develop an IVIV-based PBPK model for rats. So extensive in vitro metabolism parameters and in vivo kinetic data were developed for different pyrethroids and used in the rat model to recapitulate the in vivo pharmacokinetic results. The IVIV approach that was used successfully in rats was similarly applied to development of the human life stage PBPK model that was used to predict Cmax in adult and in children. So here, this is the PBPK model that was developed. So here is the rat model, but the human model has the same structure. But let's take a look. So the model simulates pyrethroid exposure by oral here and IV, and incorporates age-dependent rat physiology as well as maturation profiles of pyrethroids metabolism mediated by carboxylesterase and cytochrome P450 enzyme in plasma and liver. We also included a vehicle compartment to describe pyrethroids kinetics with different dosing vehicles. It's also known that absorption of a highly lipophilic compound with log P greater than 5 occurs through lymphatic vessel to a varying extent depending on the fat content in diet and the use of different oil vehicles, in addition to the usual absorption path through portal veins. So given the high lipophilicity of pyrethroids, a significant contribution of lymphatic absorption is highly probable for pyrethroids. So unlike portal vein absorption, lymphatic transport bypass hepatic first pass metabolism. So that's what you can see here. So like I said previously, we had some age-specific metabolism data for IVIVE from age-matched liver samples. So we had microsome and cytosol in vitro experiment. And all the physiology factors like blood flow and tissue volumes are already accounted for by the model. Here it's an example of the age-dependent changes in total delta-metrin metabolism. So you can see that we did some in vitro experiment in juvenile rats, so PND15, and adult rat, PND90. And you can see the difference in intrinsic clearance, which is lower in the juvenile rat compared to adults. We also have Vmax and Km for saturable metabolism. And here it's the in vivo values after in vitro to in vivo scaling. To calibrate and validate the model, we use some in vivo literature. And here you can see delta-metrin exposure in the target tissue reproduced well in different ages at various dose level. So here on the left, you have plasma concentration in adult on the top row and juvenile on the bottom row. And on the right panel, you have brain concentration in adult here and brain concentration in juvenile. The dots represent the in vivo data, and we have 3 doses. And the lines represent the simulations. And as I said, you can see that the simulations, especially around the peak of exposure, around the Cmax, which is really important because pyrethroids are more an acute exposure and the effects are acute. So the Cmax is the Cmax is of interest. So that's why the simulations are looking pretty good. We also developed a human PBPK model. Obviously, we did not have human data to validate the model, but we use the same structure as the rats. And the same approach used in rat was used in humans. So we had some human in vitro metabolism data, and all the physiology was taken from the literature and integrated into the PBPK model. We had 2 major challenges for the human in vitro studies. First, limited availability of pediatric tissue samples and human variability. So what we did is we used IVIVE with recombinant enzyme in conjunction with enzyme abundance as an alternative approach. So basically, intrinsic activity of the expressed enzyme is not equivalent to one in vivo due to differences in accessory proteins like NADPH, cytochrome P450 reductase, and cytochrome B5, but also because of different lipid environments. So it is necessary to convert the activity level of a specific enzyme in the expressed system to the activity level of this enzyme in an endogenous system. So that's what we do when we have express enzyme. And I'm not going to go into detail of this calculation, but I just wanted you to have them here. So here it's the IVIVE workflow with a recombinant enzyme. So first, we need to do a screening to basically determine which CYP and carboxylesterase are involved in the metabolism of the selected chemical. Then we do IVIVE, so we extrapolate the in vitro derived values into the in vivo for each enzyme. And then we use the abundance, which is the relative expression levels of each enzyme in the liver, and we combine with the ontogeny, which is the developmental changes in each enzyme expression and abundance across ages. And we integrate all of this to obtain a total clearance value for the given age that we can input in the PBPK model. So what about the human PBPK model structure? So the structure of the human life stage PBPK model for pyrethroid is based on the rat PBPK model. There's only two modifications compared to the rat. First, we removed the metabolic contribution of carboxylesterase to pyrethroid hydrolysis in plasma because carboxylesterase are not present in human plasma. So no significant hydrolysis of pyrethroid in plasma is expected in humans. We also removed the vehicle-specific absorption to describe pyrethroid kinetics with different dosing vehicles. Obviously because we are not doing a VAG in human, so that was logical. But for the rest, the structure is the same. We applied the same strategy. We had age-dependent physiological changes in the human population, so we created a database of physiological parameters. We also have age-dependent biochemical changes that were developed in vitro, and also age-appropriate exposure. And we did oral exposure, but also inhalation, and calculated plasma and brain concentration at different ages in the population. So, like I said, we developed a life stage database for physiological parameters. So, age-specific parameters, including body weight, cardiac output, tissue volumes, and tissue blood flows, were adapted from published life stage models. And you have examples here with body weight, where you have the body weight on this axis and the time here. And you can see the enhanced data in blue. Taken from one of the latest reports, and the simulations in orange. We have here, for example, the brain volume. The orange dots are taken from the literature, and the blue lines is the simulations. Not the simulation, but the brain equation that we used in the PBPK model. Same thing here for the fat volume. We also used in vitro data, like I said, and enzyme ontogeny for the human model development. So we had human microsomal and cytosol studies to calculate the total intrinsic metabolic clearance in the liver. We had human expressed enzyme to derive the intrinsic clearance at different ages, and human enzyme ontogeny. So here it's the example of the human enzyme ontogeny. So these enzymes are the enzymes that are used to metabolize pyrethroids. So you have carboxylesterase in macrosome and in cytosol, and also CYP, different CYP. And you can see that, especially carboxylesterase, they are almost at 100% expression of adult level at birth. For cytochrome, it depends on what kind of cytochrome, but most of them are expressed at really early ages. I think the CYP1A2 is the one that is expressed a little bit later, but they are all expressed pretty fast in life. So when we combine the ontogeny data and activity data of each enzymes, we can calculate the relative contribution of each enzyme implicated in the metabolism of pyrethroids. So here from this figure, so you can see here all the enzymes involved in the metabolism of deltametrin, cis-permetrin, and trans-permetrin, and you can see based on the color the contribution of each enzyme. So here you can see that all pyrethroids except for bifenthrin are predominantly metabolized by carboxylesterase. You can see the yellow and orange is carboxylesterase, but here you can see that carboxylesterase are less expressed. It's mainly cytochromes. Bifenthrin, so here, is only metabolized by cytochromes, and the relative proportion of enzyme contributing to metabolism does not change with age. You can see here the pie is not really changing between 6 months and 25 years old. Same thing for cis-permetrin and same thing for bifenthrin. So the total intrinsic clearance also does not change with age, which is the size of the pie. You can see the pies have the same size. So, as you remember, the FQPS safety factor to protect children was set to 3 by the EPA. Now, how do we maximize the use of available data and improve the scientific support for risk assessment? Well, by deriving a DDEF, which is a data-derived extrapolation factor, by using our life stage PBPK models. So let's look at the approach that we used. So the rat PBPK model was first used to calculate the maximal concentration at the target tissue, which is the brain, in rats at a dose equal to the internal point of departure based on neurotoxicity in rats. So that's the first step here. Then we used reverse dosimetry to determine the external dose, yielding the same internal dose as in rats. Here. Finally, forward dosimetry and Monte Carlo simulations were used to calculate age-related internal concentration from which the CSAF could be determined. So the concept of chemical specific adjustment factor, the CSAF, has been introduced to provide a method for the incorporation of quantitative data on interspecies differences or human variability into the risk assessment process. So here, let's look at this approach using an example. So we use our rat PBPK model to translate an external dose to a maximal brain concentration. So we have a rat point of departure, which was a BMDL, in adult rats at PND 90. And this point of departure for deltametrin was 1.49 milligram per kilogram. We used the rat PBPK model, single dose exposure every day, and to have a brain Cmax at steady state. And this brain Cmax was 28.28 nanogram per ml. Then we used the human PBPK model to translate this maximal brain concentration from rat, and we assumed that this concentration in brain in rat was going to be based on pharmacodynamic that was done previously, equivalent to the brain concentration in human, and we translate this concentration to an external dose. So from the human brain Cmax of 28.28 nanogram per ml, we use the human PBPK model with same thing, single dose every day to have a steady state. And we derive a human equivalent dose of 0.85 milligram per kilogram. After that, we use this 0.85 milligram per kilogram to run Monte Carlo simulation and at different ages. So here we are using an adult model, so around 25 years old. But now we can use this point of departure at different ages. So at 6 months, 1 year, 2 year, 5 year, or even 80 years old, and derive the human brain concentration and look at the sensitivity of different ages in the population. So how do we derive CSAF? So we choose two age groups and we simulate these two age groups with the PBPK model to compare the internal exposure in the most sensitive age versus the adult population. So here you have, it's a fraction. So we look at the maximal concentration in the sensitive population, which we think are children's, and we divide by the adult maximal concentration. So let's look at results. So here you have the DDF that was calculated, or CSAF. We can use these two terms. We used to run the PBPK model at the point of departure, but we also used a plus tenfold and a minus tenfold. Why? So as the EPA recommends, the consistency of CSAF values should be evaluated over a range of those surrounding the point of departure to increase the level of confidence. That's why we use the point of departure as well as a tenfold higher dose and a tenfold lower dose than the point of departure. So here we use different ages. This one doesn't really serve because 1925 it's considered as adult, but we knew that children around 1 and 5 years old were the category of ages that were the most exposed to pyrethroids. So that's why we run these two ages. And here these results indicate that no additional adjustment factor is necessary to account for age-related pharmacokinetic differences for these pyrethroids. You can see here basically the DDF, so the adjustment factor is less than 1 when we look both plasma and brain Cmax. So as a summary, life stage PBPK model was successfully developed by IVIVE for metabolism parameters and by incorporating growth physiology. The model predicted that tissue dosimetry in brain is actually lower or similar in children compared to adults, which is really different from what we were seeing in rats. And based on the use of this life stage PBPK model, the CSAF values for the pyrethroids equal or less than 1 resulted in a CSAF for age-related PK differences of 1. So the EPA actually regulated and reduced the safety factor from 3 to 1 in 2019 after the work we did on this life stage PBPK model. If you're interested, there's probably a report in the literature about this work. And multiple articles were submitted related to the rat modeling, to the human modeling, and to the risk assessment. So thank you. And if you have any question, you can reach out to me, Marjory Moreau, at Cytovation. Thank you.